In the genetic engineering laboratory, the manipulation of nucleic acids is constant, so it is essential to recognize the integrity of these to ensure their correct use in various techniques, such as PCR and cloning. For this, it is necessary to know the basis of the electrophoresis technique, which supposes an important part of the systematic procedure of the analysis, which includes separation, identification, and purification of the nucleic acids. Throughout the practice, the electrophoresis technique is performed, and a detailed analysis is made mentioning the basis of the methods to know the quality of the sample of nucleic acids when observing them in the gel. In this way, important factors to be considered are mentioned for the correct realization and visualization of electrophoresis.
Electrophoresis is a term that refers to the migration of charged molecules within a gel matrix. During this dispersion, nucleic acid fragments are separated depending on their size, shape, and charge, due to the density and porosity of the gel. This movement is powered by an electrical current that takes advantage of the negatively charged molecules of the genetic material. This technique has provided a variety of molecular analysis useful for different applications. DNA analysis. DNA is a molecule remarkable for its consistent negative charge that causes that the electrical current distributes equally in the whole molecule. Due to friction interactions with the gel surface, DNA fragments roll the gel at different speeds. In this case, the technique can be used to evaluate nucleic acid integrity or to evaluate a PCR product. Immunoelectrophoretic chamber. It is an analysis that involves protein characterization, based upon the separation of certain components being diffused against antibodies. It can be arranged in 1 or 2 dimensions. Precipitation patterns are later analyzed to determine antigen-antibody interaction. Protein analysis. Considering the different amino acid building blocks of proteins, their size can vary in identifiable patterns within gel electrophoresis. In this case, the gel is made of polyacrylamide, and the proteins to be analyzed are treated with a chemical called SDS to provide the same electrical charge to all the proteins in order to enhance a separation that is only dependent on size. Test antibiotic resistance. So as to assess induced or acquired antibiotic resistance of a bacterial strain, after DNA extraction, its genetic material can be treated with restriction enzymes that cut restriction sites found within the resistance genes. According to the patterns of DNA separation, resistance can be determined. Table of considerations. In order to make a proper nucleic acid analysis, the following parameters must be taken into consideration. Nucleic acid structure. Genetic material may have different levels of compactness, especially in the case of plasmids, and the proportion of purines and pyrimidines may also affect the traveling speed of the molecules within the gel. With this knowledge, we could have more certain expectations of the results of the gel. Matrix properties. The material in which the analysis will be run has an important effect over the results. The most common options are agarose and polyacrylamide, depending on molecular weight. Remember, concentration will influence separation resolution. Well sizes and gel thickness. An exceeded width of the gel may cause the bands to diffuse, and the well form, according to Thermo Fisher, is recommended to be long and narrow to enhance a better resolution and to avoid smears. Running buffer. For lower molecular weight samples, use TBE, and for increased molecular weights, use TAE. Running conditions. The used voltage for running the analysis is dependent on the distance between the electrodes of the device and the average size of the samples to be separated. It is important to run the analysis before the gel fuses. Dyeing solutions. Aspects like the concentration, sample affinity, toxicity, and gel penetration must be considered, because this will be the compound that will allow gel visualization. The structural characteristics of the sample must be assessed for determining this consideration. DNA-RNA gel electrophoresis. It is necessary to consider that DNA and RNA have important structural differences that may have exclusive interpretations. The capability of RNA to form secondary structures may influence molecular friction within the matrix. Thus, at the naturation electrophoresis, where the running buffer is TBE, is recommended. The dyeing agents must be chosen as the most suitable for single-stranded proteins. In some cases, RNA electrophoresis is a previous step for northern blotting. Electrophoresis principle. The principle of gel electrophoresis is the separation of sample components based upon their charge, which in turn produces that molecules travel at different speeds. In this case, nucleic acid have a negative charge and will migrate towards the positive electrode, providing the anions of the electrical field, while cations will be provided by the running buffer. The samples traveling within the gel present distinctive frictional interactions. This phenomenon is known as electroendosmosis. The mobility of a molecule is inversely proportional to its size and directly proportional to its charge. Now we present the table of the different parameters that must be taken into consideration according to the type of sample that will be analyzed.
Methodology. Make the necessary calculations to prepare the assigned agarose gel. 0.8%, 1.6%, or 2.5%. Weigh the agarose and dissolve it with 1x TBE buffer or 1x TAE in the microwave. To prepare a gel in the electrophoresis chamber, 50 ml of TBE buffer or TAE are required. Be carefully that the agarose doesn't spill in the microwave. Wait for the agarose to cool. You must be careful that the agarose not to gel. Once the agarose cool, add the gel red to 0.2x. Mix perfectly and amply into the mold of electrophoresis chamber that was previously assembled with the comb. Important! For the 2.5% agarose gel, it is not necessary to let it cool. So, put it out of the oven and apply the gel red and perform the steps as described above. Remove the comb. Cover the gel with the charge buffer TBE1X or TAE1X, registering the order of how the samples are loaded into the gel. Add the appropriate amount of loading buffer in a piece of parafilm depending on the volume that is loaded with DNA. Add the required volume of the DNA sample in the parafilm. Mix the DNA and the loading buffer with the micropipette. Add the mixed sample in the indicated well. In the first well, load 5 microliters of the molecular weight lather. Run the samples for 60 minutes at 100 volts. Remove the gel from the electrophoresis chamber and visualize in the transilluminator. For this, it is necessary to use gloves. And finally, document the image.
The loads were as followed. Rail 1, fast DNA ladder from New England Biolabs, 5 microliters. Rail 2, 2 log nano from New England Biolabs, 3 microliters. Rail 3, 100 base pairs ladder from Invitrogen, 3 microliters. Rail 4, PCR Topo 2.1 plasmid, 3 microliters. Rail 5, E. coli genomic DNA, 2 microliters. Rail 6, possum RNA, 2 microliters. And Rail 7, PCR product of 450 base pairs, 3 microliters. All of the gels were dyed with gel red at 0.2x and ran at 100 volts per 1 hour. On this first image, it's observed that rails number 1 have a better separation of the fragments in comparison to the other ladders. On the gel at 2.5%, rail 2 has a higher amount of sample. This can be detected by the irregular shape of the bands. On rail number 3 from 0.8% and 1.6% of agarose gels, there is a lack of definition of the bands. On rails number 4, from all gels, there are between 2 and 3 different bands, approximately at 4,000, 5,000 and 6,000 base pair. On the 3 agar concentration gels in rail number 5, there is a DNA degradation seen as a shadow, and a fragment bigger than 10,000 base pairs. On rail number 6, in all of the gels there is a black spot at the end, and on gels at 0.8 and 1.6% of agarose, there are also between 2 and 3 fragments, around 1,200 and 3,000 base pairs. On rail number 7, a 450 base pair fragment is appreciated. For the next gel, which corresponds to buffer TBE, 1x gels are shown in the same order as the first one, from 0.8% to 2.5% of agarose from left to right. These gels were loaded as followed. Rail 1, 100 base pairs ladder from Invitrogen 5 microliters. Rail 2, fast DNA ladder from New England Biolabs 5 microliters. Rail 3, 2 log nano from New England Biolabs 5 microliters. Rail 4, PCR Topo 2.1 plasmid 5 microliters. On gel at 1.6%, this sample was loaded twice on rails 4 and 5. Hence, gel at 1.6% has 8 rails used in total. Rail 5, E. coli genomic DNA, 4 microliters, and Rail 6 for 1.6% yield. Rail 6, possum RNA, 4 microliters, and Rail 7 for 1.6% yield. And Rail 7 PCR product of 450 base pair, 1 microliter, and Rail 8 for 1.6% yield.
Throughout the elaboration of the practice, we were able to perform an electrophoresis for the separation, identification, and analysis of nucleic acids, management of equipment and tools, as well as the correct preparation of the solutions employed in the process. Based on the concentration of agarose contained in the gel, it was possible to observe the migration of the different samples and analyze their size. The principle of electrophoresis is based on the migration of charged molecules in the presence of an electric field. In this case, the DNA, which naturally possesses a negative charge due to the phosphate groups, can migrate from the negative electrode to the positive of the chamber by applying a specific voltage, taking into account the porosity of the gel, which is directly related to the concentration of the agarose. Said migration and separation of the molecules is based on their size and shape. Additionally, it is important to mention that the running of the samples along the gel also depends on the concentration of agarose, since it modifies the porosity and density of the gel, facilitating or hindering the passage of the molecules through it. In this way, samples in a gel with 2.5% agarose will migrate less compared to samples in a gel with 0.8% agarose. Taking this into account, it is important to make the calculations to prepare a gel whose percentages of agarose is adequate for the samples to be analyzed. For this, it is possible to employ the following table that relates the different concentrations of agarose with the optimal DNA separation range. To carry out an agarose gel electrophoresis properly, it is crucial to consider the running buffer, since its performance directly affects the quantity of the results. The running buffer influences the electrophoretic mobility of the DNA molecules by providing the necessary ionic strength. That is why the gel is submerged in the buffer. In general, two different buffers are employed in the genetic engineering laboratory, TIE and TBE. Depending on the nature of the electrophoresis, the first one can be used when the running of the gel is carried out under conditions that imply lower salt content. It is more economical than TBE, but is not recommended for long runs or high voltages. On the other hand, the TBE has a higher salt content and is used for longer runs and for the separation of larger DNA fragments. Another important aspect to consider when performing an electrophoresis is the fact that the gel is completely submerged in the running buffer, which implies that the loading of the samples in the wells must be prevented from spreading in said buffer. For this purpose, a loading buffer is used, which activity consists in increasing the density of the samples and providing it with color to facilitate the visualization. Among the most employed compounds to increase density is glycerol, ficol, and sucrose, while the coloration bromophenol blue, hylencianol, and bromocresol green can be used.
In the electrophoresis performed by both groups, the marker bands could be distinguished. However, they were not distinguished with high quality. To improve the resolution of these bands, we can perform a discontinuous electrophoresis. This consists in dividing the gel in two phases. At the top, an accumulating gel or concentrator, stacking gel, which has as effect to concentrate the sample in a narrow band, and occupying most of the plate a separating gel or resulting gel, where the components separate. The concentration effect is due to the higher porosity of the accumulating gel combined with a different composition of the bucket tampons, 3-glycerin and 3-HCl, and different pH for both gels. For PCR product, sample degradation was observed because the sample was run to the lower molecular weight part of the plate. For plasma, a concentration was observed in the higher molecular weight part, indicating different levels of winding. Some protein contamination were observed in the samples, as some cells showed residues that were not released, and also excess of salt that could be observed in the distorted marks. The 6 gels show that the genomic DNA sample degraded for the most part. Only a small undegraded DNA mark could be observed. For both buffers, TA and TBA were used 3 different concentrations of agar. The concentration varies depending on the molecular weight of the sample to be analyzed. In this case, in the gel with 0.8% could be observed more clearly the separation between each band. For example, in TA, for the fast DNA marker was clearly separated in its 11 lines. However, the lines were not concentrated enough, but the gel with 2.5% agarose, the samples were not separated enough to determine their size more accurately. We therefore consider that the best arrangement for these samples was 1.5%. One of the mistakes that exists when pipetting samples is to leave samples outside the cell, or to perforate the gel with the tip. With the perforated gel, the sample does not leave the cell, or it leaves the gel just as by not depositing the whole sample inside the gel.
Agarose gel electrophoresis has been shown to be an efficient method for the separation, identification, and purification of nucleic acids. The ability to resolve nucleic acid fragments can be influenced by the matrix composition, concentration, the selection of the running buffer and the base pair ladder, the run conditions such as the voltage and the duration, the intercalating agents for the visualization of the DNA, and the quality of the sample. In order to identify and analyze several fragments of DNA of different natures, it was necessary to correctly prepare those samples and the gel, which would later be subjected to a voltage inside the electrophoresis chamber. This implies the proper manipulation of said samples at the moment of being loaded, as well as the gel when it is exposed to UV light inside the gel imaging system to be able to interpret it correctly, taking as a reference the base pair ladder, which is indispensable for the analysis of the fragments. The practice was satisfactory since both of the gels with TI and TBE in their different concentrations of agarose, it was possible to observe clearly the different bands corresponding to the ladders and to the different samples. According to the results obtained, it is possible to ensure that the most efficient ladder for the study of the fragments was the fast DNA. It was identified that the quality of the genomic DNA obtained from the E. coli bacterium doesn't have good integrity, since it was not possible to identify a defined band. This was because the sample was contaminated with RNA. The plasmid quality had a good integrity in the 1.6 agarose gel in the TBE buffer because it was the only one that presented isoforms. The other plasmid samples result in the different gels at different concentrations and with different buffers presented no isoforms. The quality of the RNA obtained from the opossum doesn't have a good integrity since it was diffused RNA is observed and the bands are not very visible. Finally, the quality of the PCR product had good integrity since it was a defined band observed in the gel made with the TBE buffer, which migrated to the expected size, which was approximately 450 base pairs.